Good afternoon, everybody. I'm Dr. Aruna, and today I would be discussing about the genetic aspects of early childhood caries. We all know that early childhood caries is a multifactorial disease, implying that it is caused mainly by an interaction between the environmental factors and the genetic factors. The role of diet and microflora as environmental factors has been well established. But the third most important component of the Keys triad, as represented by the host factors, is under total genetic control. So how do these genetic factors bring about a change in the susceptibility to disease? Well, we all know that single nucleotide polymorphisms, referred to as SNPs, are one of the most common variations in the human genome. As the name suggests, single nucleotide polymorphisms mean a change in the sequence of the base pairs along the DNA in the human genome. These changes or the genetic variations will be reflected as a change in the various traits that we observe among the individuals, such as a change in the skin color, change in the eye color, or change in the hair, or even predisposition to various disease conditions. There are many genes which can influence the susceptibility to ECC, and the studies have been conducted on these families of genes. But how do these polymorphisms bring about a change in the susceptibility? That is mainly because these polymorphisms result either in the formation of abnormal protein or no protein at all during the transcription and the translation processes, thus resulting in increased susceptibility to diseases. The most common genes which are implicated in predisposition to dental caries include those regulating amelogenesis or enamel formation, the genes regulating the immune response and the salivary fluent composition, and those determining our diet preferences and behaviors. We all know that amelogenesis is a genetically controlled phenomenon, and a recent systematic review by Dr. Kritika et al. In the year 2018 has implicated the presence of enamel defects as the strongest risk factor in the causation of dental caries. Enamel formation basically consists of three processes: matrix deposition, mineralization, and maturation. Any disturbance in any one of these three processes would result in the formation of defective enamel, which is more susceptible to acid demineralization. As depicted in this slide, the first two polymorphisms in the enamelin gene affect the functionality of the enamelin protein and affect the maturation of enamel, whereas the genetic variations found in the matrix metalloproteinases affect the turnover of matrix components and prevent the thickening of crystallites and reduce the enamel microhardness, making it more susceptible to acid attack. Polymorphisms of the immune response genes alter the defense mechanisms and either facilitate or reduce the mutant's colonization, thereby either predisposing or protecting against dental caries. We all know that saliva is referred to as the bloodstream of the tooth. The volume, the pH, the osmolarity, the protein content, the viscosity, and the buffering capacity of saliva are all regulated by a group of genes referred to as aquaporins. And genetic variations in these aquaporins have resulted in an increased viscosity of saliva, thus increasing the caries experience. Well, the sweet tooth is genetically determined. As shown in this slide, the presence of the nucleotides G, G and C, that is guanine, guanine and cytosine in these polymorphisms increases the sensitivity to taste, thus reducing the consumption of sugar and protecting against caries. But on the contrary, presence of the nucleotides C, A and T, that is cytosine, adenine and thymine in these polymorphisms would increase the insensitivity to taste and would increase the sugar consumption and predispose to ECC. So dear friends, I would like to conclude by asking you all a question. Does your DNA determine your risk of dental issues? And the answer is yes, because all illnesses do have a hereditary contribution. Thank you. These are my references. Thank you for your patient listening.